Sorry, a little bit late. New room. Make sure all the committee members are here. Our friends team. Let's deal with it. With Molly, Vic, and Chris. We have one of the committee members, Chris Langdon, on Zoom. So I'll keep it very short so we can get started. Thanks for coming in. I'm very excited and I'm excited to see you do all her research. And just a little bit of background. Andrea did her bachelor's degree at United College in Pennsylvania in 2011. And then went to get a professional science master's at Northeastern University in Boston, in marine biology. Joined OSU in 2016. Between those, she worked in some agencies like the USDA, the Newport, and the Fish and Wildlife of Pennsylvania, where she got a lot of experience doing conservation-driven research. And it's pretty clear from her topics that affected her interest in studying marine science-related climate issues and conservation while learning new genomic skills. And you will see from a seminar that the topics are very different animals, different questions that allow her to engage with.
Get all the technology to work.
Oh, it's working. Sorry, the volume on the computer is just turned
Okay, great. Thanks, Noah. Thanks, Ricardo.
All right.
All right, I'm going to continue. Is it presenting? Okay, thank you.
Sorry.
No, you're totally fine. So before I continue, oh, sorry, I didn't even read my intro slide. So thank you all for coming. Today I'm going to be presenting my defense titled Adaptive Potential of Coastal Invertebrates to Environmental Stressors and Climate Change. But before I get into it, I want to acknowledge the peoples in which land I have conducted my research on, the Kayupuya tribes. The Kayupuya people were forcibly removed in Western Oregon several years ago. Fun fact, the Kayupuya tribes, they had established gender roles, but females who were considered wise were held in high esteem. And for this reason, some of them can actually be leaders within their bands, which I think is pretty cool. So as someone who has lived and worked on these lands for the last 6 years, I want to recognize and respect the Kayupuya people, past and present. Okay, so diving in, global climate change due to anthropogenic stressors or anthropogenic carbon emissions has negatively impacted our oceans in a variety of ways, as carbon dioxide has increased by 48% since the Industrial Revolution. This is concerning for our oceans, which act like a sponge, absorbing roughly 30 to 48% of these carbon emissions. As a result, absorption of carbon dioxide results in oceans being more acidified, warmer, and also having lower oxygen concentrations. The IPCC has provided reports predicting how these different climate stressors are going to look by the year 2100. And so in the next 3 slides I'm going to show you, there's going to be these figures where the blue shows projections of what the what it's going to look like by the year 2100 if we were to not reduce our carbon emissions. So it's kind of the worst case scenario. And then the orange represents if we were to decrease our carbon emissions. So I'm going to report things based on this range. So looking at this first figure for sea surface temperature, we see that temperature is going to increase anywhere from 1 to 3 degrees by the year 2100. Warmer waters has resulted in many organisms having a wide range of physiological effects. But what I'm going to focus more on is that warmer temperatures are more preferable to pathogens who thrive in these conditions. In addition, hosts can be more susceptible to these pathogens as they may be thermally stressed. So we have seen that marine diseases have increased over the last few decades. Absorption of carbon dioxide also results in sea chemistry shifts that results in these oceans becoming more acidified. We have seen the pH of our oceans drop from anywhere from 8.2 to 8.1, with projections predicting that it's going to further drop from 8.05 to 7.75 by the year 2100. This is concerning for calcifying organisms who have a harder time building their calcium carbonate shells within these acidified conditions. Lastly, warmer waters make it so that way oxygen is not held as well in the ocean, and so we are seeing our oceans are becoming less oxygenated. It is predicted that oxygen concentrations are going to drop 1 to 4% by the year 2100, with variation across different regions. So for instance, off of our coast, we are seeing that oxygen concentrations are dropping so low that we are seeing more events of hypoxia, especially during upwelling seasons when these hypoxic conditions get upwelled and are exposed to organisms off of our coastline. So because of climate change, these three climate stressors are heavily studied when trying to understand how marine organisms are going to be impacted and adapt to these different climate stressors. However, there still are a wide range of gaps in our knowledge about how these different climate stressors are going to impact many species, including those that are economically and ecologically important to the United States. So in my dissertation, I assess how 3 different species are impacted and may adapt to these different climate stressors. So first, I assess the genetic differences in sick and healthy sea stars during the sea star wasting epidemic within the keystone species Pisastereocrasis. Next, I examine differences in gene expression in the Pacific oyster during their settlement when exposed to acidified conditions, which is another economically important, is an economically important species to the United States. And then lastly, I estimate heritability of metabolic processes in the red abalone haliotis, another economically important species, when exposed to these OA or hypoxic conditions. For all 3 of my chapters, I will utilize these icons to demonstrate which species I am discussing. So starting off with my first chapter titled Little Evidence for Genetic Variation Associated with Susceptibility to Sea Star Wasting Syndrome in the Keystone Species Pisastereocrasis. In this chapter, we look at whether or not there is any genetic composition that allows Pisaster to be more resistant to sea star wasting. So in 2013, the sea star wasting epidemic decimated over 20 species of sea stars ranging from Baja California, Mexico to the Gulf of Alaska. Of the species impacted included that of the keystone species Pisastereocrasis, who saw populations decline ranging from 50 to 94% in different areas in Oregon. This is concerning as they are keystone species, meaning that the loss of these populations could have downstream effects on the intertidal habitats in which they live. Sea star wasting presents itself as tissue lesions, loss of grip strength, arm torsion, tissue degradation, and then also high rates of mortality. The causation of sea star wasting is still widely debated, with hypotheses ranging from a viral infection to a stress response from shifts in environmental conditions. However, comparison of disease severity across different regions has often seen that areas with warmer water had higher rates of disease severity and mortality. For example, in Eisenlord et al., they observed sea stars within several sites within Washington state, and then used this data to model the probability of disease over time as a function of temperature. From this, they concluded that for every 1 degree increase in seawater temperature, the probability of disease increased by 1.4%. This is concerning as we know that seawater temperatures are going to continue to increase, potentially influencing future outbreaks, leading to questions of not only if, but when the next outbreak will occur and whether or not sea stars, including that of Pisaster, will be able to survive. So if Pie-Zaster has the genetic composition in order to reduce their susceptibility to C-star wasting, then this could provide some insight as to whether or not they can adapt to future outbreaks. Shibboleth et al. Addressed this question by performing a discriminant analysis principles component test, for DAPC for short. I'm going to first explain what this methodology is. And so what DAPC does is it performs a principles component test prior to estimating density clusters based on individuals' genetic composition. The end result is these density clusters with two populations represented by peaks. And if the peaks are overlapping with one another, this means that these populations are genetically similar. Whereas if these population peaks are separated, this means they're genetically distinct or had differences in the genetic composition. So looking at Shibboleth et al. Study, they sampled Pie-Zaster before the outbreak in 2012, and then again after the outbreak in 2013. And looking at their DAPC analysis, we see that there was an allelic shift before and after the outbreak, suggesting that surviving Pie-Zaster may have some reduced susceptibility to C-star wasting. However, because they sampled before and after the outbreak, basically at two different time points, it makes it unclear as to whether or not other environmental factors may have attributed to the differences between these two disease statuses. In addition, they sampled Pie-Zaster only in California and may not be representative of the entire species across their geographic range. So in our study, we build upon Shibboleth et al.'s work by comparing sick and healthy Pie-Zaster sampled during the peak of the C-star wasting outbreak that were sampled within Oregon. So before I continue, I want to clarify, I will be referring to individuals as either asymptomatic or symptomatic to take into account sick individuals who had yet to exhibit symptoms at the time of sampling. So in 2016, a team of scientists from Oregon State went out and sampled Pie-Zaster from 6 different sites in Central Oregon. The advantage was that we sampled during the peak of the C-star wasting epidemic, in which asymptomatic and symptomatic Pie-Zaster were co-occurring either in the same or adjacent tide pools. So this removed any uncertainty as to whether other environmental factors attributed to differences in their disease status. During sampling, Pie-Zaster were given a disease rank based on Menge et al.'s disease ranking protocol, in which 0 represented asymptomatic individuals that had no lesions. And then the disease rank increased based on disease severity, with 6 being the highest disease ranking value in which we saw tissue degradation. From these samples, we then took a subset of 112 asymptomatic individuals and 82 symptomatic, of which only consisted of those with the highest disease rank in order to maximize the power between asymptomatic and symptomatic individuals. From these samples, we then extracted DNA and prepared their libraries using a 2B-RAD genotyping protocol. Sequence reads were then processed and mapped to a reference genome for Pie-Zaster loquacius. In the end, we ended up having around 71,000 cleaned reads for processing or for analysis. We then used 2 different types of analyses in order to compare disease severity, asymptomatic and symptomatic individuals. The first being a base scan analysis that allowed us to find outlier FSTs that are indicative of loci with a strong effect. And the second being the same DAPC analysis we saw from Shibboleth et al. All study in order to look for genomic regions with some small contributions to differences between disease status. So moving on to the results, let's first look at the base scan analysis. For our base scan analysis, we found 0 outlier FSTs, and FST measures the amount of genetic divergence within or between populations. And so a high FST means that these 2 populations are going to be genetically different from one another, whereas a low FST means that they are more genetically similar. So looking at our FST estimates for all 70,000 SNPs comparing asymptomatic and symptomatic individuals, these FSTs are represented across the genome, with each chromosome represented by alternating red and gray coloration. In addition, we obtained p-values in order to determine significance for each one of the FST estimates. And then this dashed line indicates the lowest adjusted p-value. So from this, we see that all our FST estimates were all fairly low, and none of them were significant, meaning that there was no single locus that attributed to differences between asymptomatic and symptomatic individuals. Moving on into the DAPC analysis. Within our figure, we see that there's quite a bit of overlap between asymptomatic and symptomatic individuals. However, we do see some slight separation between the 2 disease statuses. We were then interested to see what genomic regions may attribute to the modest separation between these 2 groups. So for this, we pulled out the loading values from the DAPC analysis for each locus. A loading value essentially just measures how much that single locus contributes to that separation between the two disease groups. So from this figure, we drew a line separating the visibly larger loci loading values from this, and which allowed us to find 18 SNPs that had the largest contribution to the differences between the two disease statuses. We were then curious to know where these loci were located on the genome and if they had any adjacent protein coding genes. But to do that, we first needed to figure out where the cutoff was for recombination is in Pisaster. So to do this, we estimated linkage disequilibrium between pairs of SNPs known as R squared. And then we averaged the R squared within bins of 100 base pairs, which is represented by all these points. In this figure, we see that there's quite, the linkage disequilibrium declines fairly rapidly until around 15,000 base pairs, meaning that any marker and protein coding genes found within 15,000 kilobases from one another is likely linked. We then use this parameter to then look for associated protein coding genes. So for example, from the DAPC analysis, we've zoomed in on one of the SNPs that we found, and we looked plus and minus that 15,000 kilobase range in order to find protein coding genes shown in yellow. This allowed us to find 34 protein coding genes that may have some contribution to differences between symptomatic and asymptomatic individuals. In addition, we compared our markers found from our study to that from Shibboleth et al. To see if we found any similarity in our genomic regions. However, we did not see any similarity in genomic regions across Shibboleth et al. And our study. The lack of similarity between Shibboleth et al. And our study may come down to just how different our methods were performed. For example, we sampled in Oregon, whereas they sampled in California. During the year sampling, Oregon had experienced more days in the year in which the seawater temperature had deviated from the norm, meaning that populations in Oregon may have experienced slightly different selective pressures than that in California, and then thus may have different genetic compositions. So in conclusion, our study found that there's little genetic basis for reduced susceptibility to C-star wasting, as we found no single locus that attributed to differences between asymptomatic and symptomatic individuals. In addition, we didn't really see a large separation within our DAPC analysis. However, the genomic regions and functional candidates that we found from the study could provide some insight into future studies when trying to understand reduced susceptibility to C-star wasting. But having said that, it is likely that Pisaster are going to be susceptible if there are future outbreaks as climate change continues to increase our seawater temperatures. And lastly, due to the lack of similarity between Shibboleth et al. And our study, we suggest that future studies should look at a wider geographic range, as the genetic composition may differ within different species, and some populations may have adaptive potential that we couldn't measure in Oregon. Okay, moving on to my next chapter, titled Assessing Ocean Acidification Impacts on Gene Expression During Settlement of Crasastia jagus. In this chapter, we look at whether or not ocean acidification impacts gene expression during settlement within Crasastia jagus. So in this figure, we see examples of some of the more commonly studied organisms when understanding the impacts of ocean acidification, including echinoderms, bivalves, including Crasastia jagus and coral, with their different larval stages separated by those in an ambient or normal seawater conditions. So these are healthy larvae, and then two different acidified conditions. From this image, we can see the impacts of ocean acidification on these organisms. So for example, we see that there's reduced calcification. We see cracks or corrosion in the shell, malformed shells, dissolution, and then also just ocean acidification results in high mortality. Early larval developmental stages are really vulnerable to these acidified conditions due to having a high surface area to seawater ratio, in addition to the metabolic demands of trying to mitigate these acidified conditions during such energetically demanding developmental stages. So for example, larvae first build their calcium carbonate shell after a few hours after fertilization, which is energetically taxing. As a result, in acidified conditions, we see that they have malformed shells and can even be reduced in their size. Larvae can also be developmentally delayed, resulting in them not reaching metamorphosis at quick of rates as those in ambient or normal conditions. And then also settlement can also result in having malformed individuals or juveniles along with having reduced size. In Crustacea aegagus, there are 4 stages in development prior to becoming a juvenile. First, we have a fertilized egg that quickly develops into a dehinged larvae. Over a few days, this dehinged larvae develops into a veliger larvae. And then in latent stages, this larvae will develop an eye spot, at which point we refer to it as a pediveliger larvae. This eye spot is important because it indicates that this individual is then ready to undergo settlement and a metamorphosis into a juvenile spat. In natural conditions, these larvae will settle onto a substrate prior to undergoing metamorphosis. However, in aquaculture settings, the hormone epinephrine is utilized in order to induce metamorphosis, but as a result, some individuals will bypass settlement. Thus, they will undergo metamorphosis while still in the seawater column. In this study, it is important to distinguish between these 2 outcomes. So I will refer to individuals that bypass settlement as unattached spat, and then those that had cemented themselves and undergone settlement onto a substrate as attached spat. So metamorphosis is an energetically taxing process that has been heavily studied when looking at the impacts of the ocean acidification. However, less studied is that of settlement, which requires that this individual secretes a calcium mineral deposit onto a substrate before it can undergo metamorphosis. The transition from a pediveliger larvae to a juvenile spat happens really rapidly, including these 2 processes happen asynchronously, but one right after another, making it difficult to parse out the period in which these processes happen. For this reason, settlement and metamorphosis are often used interchangeably within the literature, making it difficult to assess how ocean acidification impacts one or both of them. So in our study, we hone in on genes that are related to settlement that are impacted by acidified conditions. To test this, we spawned and reared larvae over a 22-day period in which individuals were undergoing settlement and metamorphosis towards the end of this experiment. Larvae were obtained by stripping and cross-fertilizing eggs and sperm from families in which we obtained them from the molluscan broodstock program. These individuals were maintained in either an ambient or high PCO2 treatment, which was manipulated by adding carbon dioxide until the desired pH was reached and then allowed to aerate overnight. Larvae were first exposed to these different conditions roughly 5 hours after fertilization, at which point we had standardized them from their known families and pulled them before putting them into these treatments. They were maintained in the same PCO2 treatment throughout the experiment, with water changes occurring every 2 days in order to maintain and measure seawater chemistry. On day 12, larvae were separated into their experimental replicates with 4 replicates per treatment, along with having 5 days of sampling every other day in order to obtain count measurements to measure percent metamorphosis and settlement. On day 14, we began sampling, as this was the first time we observed pediveliger larvae, indicating that these individuals were ready to undergo settlement and metamorphosis. On the final day of sampling, we subsetted our cultures in order to have some of these cultures ready for count measurements along with transcriptomics. So in natural conditions, competent larvae are going to settle and metamorphose over a wide range of timescale, making it difficult to sample at a single time point because you're just not going to have enough individuals for measurements. For this reason, we utilize the hormone epinephrine in order to induce competent individuals to settle and metamorphose, narrowing that time period. But we were concerned about the interactive effect of epinephrine on PCO2 treatment. For this reason, during the days of sampling, we split our cultures in half in order to expose half of the individuals to epinephrine, while the remaining were not exposed to epinephrine. They were left alone for 2 hours while the epinephrine took effect, at which point we then subsetted these cultures in order to have around 1/4 of the cultures dedicated to preserving veliger, pediveliger, and unattached spat for count measurements. Attached spat that had cemented themselves onto the jar were then counted separately. We use the total number of individuals then to calculate the percent metamorphosis, consisting of unattached and attached spat, and then also percent settlement of just having attached spat. On the final day of sampling, where we also subsetted these cultures for transcriptomics, the method started off exactly the same way, so we could still obtain those count measurements. But because we subsetted these, we had leftover cultures in order to preserve petty villager and unattached fat in RNA later for transcriptomics, which I will refer to as non-settled individuals. Attached fat I had cemented onto the wall were quickly counted before then scraping them off and preserving them separately. Due to a lack of epinephrine exposure in the control treatment, we did not have any attached fat that were exposed to epinephrine or not exposed to epinephrine. Sorry about that. So in the end, we had 3 samples per PCO2 treatment across all replicates, including non-settled individuals exposed to epinephrine, non-settled individuals not exposed to epinephrine, and then attached fat, all of which were exposed to epinephrine. These samples were then prepared using a tag-based RNA-seq method. These reads were then processed and mapped to a gene model for Chrysastria gigas that was derived from a reference genome for Chrysastria gigas. Statistical analysis was then done with EDGE-R, and then Go terms were relating to differentially expressed genes were enriched. So Go terms or gene ontology terms, these are derived from a database, a standardized database that provides terms that kind of give a functional description of what these different genes may be related to. So first, for the first model, we looked at whether or not epinephrine had a interactive effects with PCO2 treatment. So to test this, we performed a contrast model comparing high PCO2 to ambient PCO2 while taking into account individuals exposed to epinephrine and those that were not. This model resulted in 0 differentially expressed genes, meaning that there was not an interactive effect with epinephrine and PCO2 treatment, meaning any downstream analysis where we did utilize individuals exposed to epinephrine wouldn't result in differentially expressed genes due to that hormone. So then we looked at the differences in gene expression in PCO2 treatment within non-settled individuals and attached fat separately. And then lastly, we looked at genes that were related to settlement through this contrast model, comparing high PCO2 to ambient PCO2 treatments while taking into account attached fat and non-settled individuals, kind of like before and after settlement. So moving on into our results, first looking at that count measurements. Metamorphosis, looking at percent metamorphosis, we see that there's no differences between ambient and high PCO2 in the first 3 days of sampling. However, on days 20 and 22, we see that ambient conditions had higher rates of metamorphosis, suggesting that larvae reared in the high PCO2 treatment were delayed and thus weren't competent to undergo settlement. This is not uncommon, has been observed in other studies, and thus is not surprising. However, when looking at percent settlement, we don't see any differences between the PCO2 treatments on any day of sampling, suggesting that these individuals may not be, the outcome of settlement is not impacted by acidified conditions. This has been observed in other studies, such as Durlin et al., who also saw that percent settlement was not different between ambient and high PCO2 treatments in 2 different cultures consisting of individuals from the molluscan broodstock program and also wild populations from Willapa Bay. Okay, so moving on to the transcriptomics. Here is a heat map. I'll break it down for you. Each column represents a replicate separated by the ambient and high PCO2 treatment, and then further separated by attached fat and non-settled individuals. Each row is a single gene's expression level, where the expression level has been scaled based on high expression in red, white is moderate, and then blue is low expression. And these different genes have been separated based on their Go terminology of biological, molecular, and cellular. So to break this figure down, let's first look at non-settled individuals. Within this model, we found 453 differentially expressed genes that were largely upregulated in the high PCO2 treatment, which makes sense when we look at our heat map, where we see consistency across the replicates, where this single gene has high expression levels across all 3 replicates. But when we look at attached fat, we found 0 differentially expressed genes. When looking at the heat map, we can just see that there's a lot of variation across the replicates, where many of them have moderate gene expression, but for example, this replicate here had high expression levels, and then 2 of these other ones had low expression levels. So the amount of variation across these replicates explains why none of these genes could be deemed statistically significant, resulting in 0 differentially expressed genes. It could be that these acidified conditions just doesn't have a very strong effect on individuals post-settlement. which is kind of consistent with what we saw when we looked at settlement rates. However, carryover effects from juvenile to, larvae to juveniles within acidified conditions has been observed. In our experiment, we sampled juveniles directly after settlement. It could be that we just sampled too soon for any of these carryover effects to really present themselves. Follow-up studies looking at differentially expressed genes in juveniles in increments post-settlement could kind of tackle this question. So moving on to the main purpose of the study, looking at differentially expressed genes associated with settlement process. In this model, we found 943 differentially expressed genes. Enrichment from these genes resulted in GO terms that are 31 GO terms that can broadly be categorized into these 8 functional classifications. The majority of the genes relating to these GO terms were downregulated in the high PCO2 treatment. When conducting a literature review in order to understand what these different GO terms may, the purpose of them within Crasastia jagus, there's not a lot of consistency across the literature, and basically they're poorly understood within Crasastia jagus and other marine invertebrates as well. However, these 3 GO terms were seen in similar studies looking at gene expression in different marine invertebrates, including echinoderms, mussels, and some other oysters as well, within acidified conditions. These studies suggest that these genes relating to these GO terms are important for settlement and calcification, which makes sense for settlement because that's what we were looking for. And calcification also kind of confirms because these individuals are producing a calcium mineral deposit while they're undergoing settlement. So in conclusion, we found that there is maybe no impact of acidified conditions on individuals post-settlement. This suggests that juveniles may be less vulnerable to ocean acidification as they will be able to survive within these conditions, potentially driving adaptation as they produce their own offspring in this environment. We found several genes relating to settlement that are impacted by acidified conditions. These functional candidates provide further insight as to how ocean acidification is going to impact larvae throughout their development. Lastly, the complexity of physiology across the different developmental stages highlights the need to distinguish between settlement, metamorphosis, and other developmental processes when trying to understand the impacts of these climate stressors on throughout the entire development of the oyster. Okay, last chapter. Titled Estimating Heritability of Variation in Respiration Rates in Heliodystropheans. In this chapter, we look at whether or not red abalone have the capacity to adapt to future OA conditions by estimating heritability in metabolic processes. So in the last chapter, I introduced before that acidified conditions are going to be concerning for calcifying organisms. Off of our coastline, hypoxia is also a concern due to seasonal upwelling. This is because our oceans are striated, in which deeper layers have respiring organisms that contribute to carbon dioxide, making these deeper layers more acidified, in addition to taking up what oxygen is there. So these deeper layers are less oxygenated than these top layers. Climate change is amplifying these processes where ocean acidification is making these deeper layers more acidified than in the past, in addition to warmer temperatures making it so the ocean holds less oxygen. And so these deeper layers are now reaching hypoxic conditions more frequently. For this reason, ocean acidification and hypoxia are not mutually exclusive, as both of these processes are occurring in these deep layers. And so during the summer months, when we have wind-induced upwelling, organisms off of our coastline are exposed to these OAH conditions. Metabolic processes are a key trait of concern when trying to understand the impacts of hypoxia on organisms. For example, Deutsch et al. Used metabolic indices in order to estimate the vulnerability of several different species to hypoxia. However, this study, like many others, only focuses on one of these components, where, again, OAH is not mutually exclusive when trying to understand the adaptive potential of organisms to future climate change stressors. These need to be considered together, as it is going to be both of these exposed to the organisms at once. So for example, in red abalone, we know very little about the impacts of ocean acidification, hypoxia, or the synergistic effects together on this organism, which is concerning because they are a economically important species due to being a source of protein, along with being culturally iconic as people like to purchase their shells. Red abalone were once abundant, but due to overharvesting, they have since experienced population collapses and are now scarce. If red abalone have the genetic capacity in order to pass on traits relating to metabolic processes from one generation to the next, they might have the potential to adapt to future climate stressors. To measure this, we estimated heritability, which partitions how much of the phenotypic variation is attributed by the additive genetic variation, which essentially just kind of gets at how much of their genetic composition can be passed on from one generation to the next. So to estimate heritability, I collaborated on a project with Hannah Gosner, who for her thesis work measured the respiration rates as a proxy for metabolic processes of red abalone exposed to 4 OAH treatments, consisting of pre-industrial, baseline meaning current OAH conditions, contemporary is near future OAH conditions, and then distant future is the worst case scenario by 2100. She had exposed these larvae to these conditions and measured their respiration rates. And we can see from her results that the pre-industrial scenario had the highest respiration rates. And then the baseline, contemporary, and future, so current and future upwelling scenarios, these were not statistically different from one another and all yielded very low respiration rates. She concluded that red abalone are currently vulnerable to OAH scenarios. However, within her experiment, she saw that there was a lot of variation in respiration rates within each treatment, suggesting that phenotypic variation may drive adaptation if traits relating to these processes can be inherited, which is where my end of the project came in. But to take a step back, I want to explain Hannah's methods. So I obtained the same red abalone that she used for this experiment. Within her experiment, she had these 4 OAA treatments where she manipulated the seawater by adding carbon dioxide to get the desired pH, and then added oxygen through agitation and diffusion. We had obtained our red abalone from Moss Landing, and these juveniles were the result of crosses in which parent females all came from one abalone farm, and then adult males all came from another abalone farm. As a result, the juveniles that we obtained were largely unrelated. Around 28 juvenile larvae were then exposed to one of the OAH treatments, during which time respiration rates were measured for about an hour, with the results being seen here from Hannah's thesis. I then obtained these red abalone and extracted their DNA. These libraries were then prepared using a 2B rad genotyping method. Sequence reads were then processed and mapped to a reference genome for red abalone, and then genotypes were called based on the nucleotide frequency at each position. Traditionally, heritability was estimated from known pedigrees in which the relatedness amongst siblings, offspring, and parents are known. For example, in half sibs who share a single parent are going to have roughly 25% similar genetic composition to one another. However, maintaining multiple generations in order to obtain these known pedigrees is not applicable to all organisms. For this reason, SNP-based heritability methods have been utilized in order to estimate the genetic relatedness seen in this matrix here between individuals. For example, individuals A and B, based on their SNP composition, had roughly about 0.42 similarity to one another, so making them largely related, whereas individuals A and C are not as related to one another. There are several different relatedness estimators, of which use slightly different modifications of how they estimate relatedness. So first, we wanted to look at which of these 6 relatedness estimators provides the most accurate relatedness estimates for our data set. To do this, we ran a simulation with using our individuals' allele frequencies within these known groups. The result being these box plots where the relatedness estimate is shown for all 6 of the relatedness estimators. The asterisk within this figure indicates relatedness estimates that fell within 0.001 of that known relatedness value. So for example, again, half sibs having relatedness around 0.25. Lynch and Lee, Wang, and Quayler all had relatedness estimates that fell within 0.001 of that 0.25 parameter. So looking at these simulations, we found that Lynch and Lee and Wang, which I refer to as L and W, all had relatedness estimates that fell within 0.001 of that parameter for 3 of the 4 simulations. So for this reason, we used LL and W in order to estimate our genetic relatedness matrix using the package related in order to obtain our additive genetic variation. We then used a restriction maximum likelihood or REML model in order to obtain the phenotypic variation from Hannah's respiration data using the package regress. These two parameters were then used in order to estimate heritability using the package gap. Our first run through resulted in many of our models not estimating heritability. After looking through some literature, we determined that it was because our matrices, our relatedness matrices were largely non-positive definite, meaning they had a lot of negative values, making it so that way our REML models couldn't estimate a coefficient. Yang et al. Explains that having a non-positive definite matrix can be the result of having too few loci in which your power is reduced, or having individuals who are largely unrelated, as these are just negative values in which they are not related to one another, which is the case for our data set because we had around 300 loci that studies suggest you should have around thousands, especially if you have unrelated individuals, which our population was outbred to be unrelated. So for this reason, we used some additional steps in order to improve our data set, including removing samples with high confidence intervals in order to ensure that the individuals that we were using were more reliable on their relatedness estimates. We then also removed loci with minor allele frequencies less than 0.05. A minor allele frequency is the frequency in which the second allele occurs within a population and has been shown to bias heritability estimates. Using these additional parameters, we were then successfully able to estimate heritability across all of our models. So I first wanted to show this figure, which was replotted from Hannah's data that I talked about in the intro slides, but I wanted to show it again as it is the phenotypic variation that was used to estimate heritability. But looking at our heritability estimates, these across the LL and W relatedness estimators, first I want to point out that the pre-industrial treatment resulted in negative heritability estimates. And this is because when looking at the amount of phenotypic variation, it's very large, meaning our denominator is also really large, resulting in a negative heritability estimate. But when looking at the baseline, contemporary, and future upwelling scenarios, we all found positive heritability estimates, with the highest being occurring within the contemporary upwelling scenario, in which LL and W averaged to 0.24 for heritability of metabolic processes. However, caution should be taken when inferring this heritability estimate for other populations, including wild red abalone, as populations are going to have slightly different genetic composition, or are going to have different genetic composition, resulting in different heritability estimates. However, our study does suggest that within our population of red abalone from an aquaculture setting, there are traits relating to metabolic processes that can be passed on from one generation to the next. So in conclusion, we found first that respiration rates were lowest in the baseline, contemporary, and future upwelling scenario, possibly making red abalone susceptible or vulnerable to current and future upwelling scenarios. However, we did provide evidence that these metabolic processes, traits relating to these metabolic processes, can pass on from one generation to the next, potentially driving adaptation if these traits are selected upon in these OAH scenarios. However, we did run into some hurdles when trying to estimate heritability. And so some suggestions for future studies is to have a larger population size, along with taking into consideration the characteristics of the population in order to ensure that there is some relatedness. You want variation within your population in order to have more accurate heritability estimates. So in conclusion, looking at these three different species under these different climate stressors, first, we see that Piesaster had little genetic basis for reduced susceptibility to sea star wasting, making them likely vulnerable to future outbreaks if they were to occur if temperatures continue to, or temperatures potentially drive another outbreak. Within Chrysastia dragus, we found several differentially expressed genes that are related to settlement that are impacted by acidified conditions. We also saw that there was no impact on individuals post-settlement within these acidified conditions, suggesting that these juveniles are likely to survive in these acidified conditions and may drive adaptation as they produce offspring for themselves in these conditions. And lastly, we saw that respiration rates were really low in current and future upwelling OAH scenarios in red abalone. But we also saw that traits relating to these metabolic processes can be inherited from parents, and so they may be drive adaptation as the traits are selected upon. So all in all, we have 3 chapters that contribute to a growing body of literature trying to understand how different climate stressors are going to impact marine organisms. However, there are still many organisms which we have no baseline knowledge about the impacts of climate change or these different climate stressors. But utilizing methods within these different chapters can provide some insight as to how to assess adaptive potential of these species when thinking about how they're going to be impacted by climate change. Okay, so I first want to thank my committee members. You all have been super amazing in just helping me throughout the years, really helping me learn. I want to give a special thanks to Eli, who helped me start out my career and helped me develop all these chapters. But I want to give a special thanks to Felipe, who made my transition into his lab super easy. I can't imagine it. I was really scared at first. I didn't know how I was going to feel about transitioning to a new lab, but you made it so easy. And I've learned a lot from you, and I really, really appreciate everything that you've done for me over the last couple of years. I would like to thank Tara, Teresa, and Trudy for keeping track of all the things that tend to slip my mind, like signing up for credits, helping me get all of this stuff set up, and then also providing me keys whenever I accidentally lock myself out of my office on several occasions. I want to give a special thanks to Lindsay, Lori, and Devin. You all have been the most amazing mentors and instructors over the last couple of years. I've learned so much from you, and I really aspire to be just like you as I move forward in my career. I want to thank Evan and Sarah for being just really amazing friends, along with helping me develop 2 of my chapters. And I want to give a special thanks to Dan and Katie, who I will dub my secondary advisors, for being just really approachable friends that I could ask questions and go over code with whenever I was too embarrassed to ask my committee members. I also want to thank my lab mates, Maureen, Chris, Cameron, and Holland, for providing continual insight into developing PowerPoints, defenses, everything, reading papers. It's unfortunate that some of us didn't get to know each other very well during the pandemic, as we were all isolated, but I really appreciate how we've been trying to make up for lost time over the last few months. And so I encourage you to continue your weekly writing meetings. I would also like to thank all my friends who I've made throughout the years for providing just continual diversions from grad life, giving me some mental breaks from what can be kind of really stressful. So thank you for the game nights, floating the river, just hanging out, hiking, backpacking, etc. I want to thank my partner, Marshall, where, verbal sentimentality isn't really my thing, especially in public settings. So instead, I just found a bunch of embarrassing photos. But kind of on a serious note, this last year should have been incredibly stressful for me, but I honestly never really felt that overwhelmed. And I think you had a major role to play in it. So thank you. I also want to thank my amazing family. I truly won the lottery with all of you. You have been my biggest supporters throughout the years, despite the fact that I have a tendency to just go off and live in another country for a year, making you have to cat sit for me. Sorry for that. My brother, Andrew, has been my first and biggest role model throughout my entire life. As for his love for science, nature, and animals is definitely a big reason why I decided to become a biologist myself. Andrew took time out of his really busy double life of working as a nurse during the pandemic, in addition to finishing up his master's degree, to draw the icons that you saw here today. So thank you so much for that. I love you all, and I constantly aspire to continue to make you proud. So thank you. Any questions? Oh, goodness.
So just procedurally, want to get out of here in a few minutes because we have the final exam with the committee. So I'll ask the committee to save their burning questions for the exam. Need them next hour. But general audience, you're welcome to entertain questions now for 2 minutes.
Yeah, Jeannie?
I'm curious, in your C-STAR chapter, are you at all concerned that your asymptomatic individuals might include both individuals who had never been exposed and individuals who were resistant?
So you're asking first that if any of the asymptomatic individuals were not exposed
Yeah.
to C-star
That
leucine?
happened.
So I personally didn't sample these, but from my understanding is no, not for the most part. Maybe not every individual was exposed to potential pathogens that may have resulted in that, but we did sample some of the individuals that were in co-occurring pools or in an adjacent pool where at some point that seawater had mixed that stuff around. So for that first part, no, I think most of them probably were exposed to whatever pathogen or causative agent may be attributed. And what was your second part?
I don't, that there wasn't really a second part. I was thinking that it might be interesting to compare like the degree of symptom, like 1 to 6, for example, because
Right.
you know they've been exposed, but they're not showing,
Mhm.
they're not as symptomatic. But I don't know anything. I mean, maybe they don't ever, maybe they always progress. I don't know anything about the disease. So
My understanding is for the most part, most of the time it does progress, but there are instances in which less severe cases such as just having like tissue lesions, they can get better. And so there are examples of people studying recovering, but we didn't look at that.
okay.
Yeah.
Cool.
I have a question about epinephrine. Would those individuals create epinephrine, or is it present in the water? Like, are there other organisms that are releasing epinephrine to cause that settlement? Or is that just an artifact of, we figured out this works in the lab, so we're going to use it for this purpose?
Yeah, so your question is whether or not epinephrine can occur within nature. So this has been,
We know it occurs in
sorry. This has been actually something that's used a lot in aquaculture, which is why we often utilize it. On many studies that you look at with looking at larval development in oysters almost always use the hormone epinephrine because it's used in aquaculture settings so much. So I don't believe that any of them, like you could just find it in the seawater column, but it is something I know that we often induce in aquaculture settings and also for these types of experiments. Yep.
So I have a question about the C-star chapter, of course. So the Shiloh paper sampled the adult populations of healthy individuals before and after.
Yes.
And so, those individuals that they sampled had, ostensibly like being exposed to kind of like a cumulative timeline of disease before and after, whereas the ones that you sampled were at the same time, snapshot,
Mhm.
healthy and diseased. And, the ones who were, that happened like 2 years after the disease began. And so I'm just curious about the timeline of what those animals, what the difference is between those animals you sampled versus the difference between the ones that Lauren did, and how you think that the timing of the sampling and the fact that, the ones that were asymptomatic when we sampled could have gotten sick the next week,
Mhm.
right? How that changes how you think about why Lauren found differences and you didn't.
Yeah, I've been wondering about that, and I wish I had access to the whiteboard at the moment. Where I kind of picture this graph where you have time on the x-axis and you see genetic differences on the y, and I kind of picture this asymptote where it kind of levels off, and so it's Lauren might have sampled really early on, and so that's where they were able to measure those genetic differences, but then we sampled later where it kind of leveled off. And so that's another thing that I think might have attributed of many other factors that could have resulted in why we didn't see differences, is that we were kind of measuring them at the tail end of their maximum genetic differences to have reduced susceptibility.
Right.
Yeah. There was more to your question. Did I get it all?
You got it.
Okay.
There's a time component in there that
Yeah.
is tricky to disentangle.
Because we sampled 2 years after them.
Right.
Bruce.
Are your data for supposedly normal animals comparable to, can you compare them to what Shebel got in terms in the before animals? In other words, I'm interested if there are genetic differences between the Oregon and the California populations. Can you look at your data in that way?
We didn't do that, and it would be curious to know whether or not we could directly compare our genotypes from our study from Menge et al. that would be interesting to look at. My first assumption would be populations are probably going to be genetically different as they are really far apart along the geographic scale. But no, we didn't look into that. It'd be cool to look into.
Mhm.
Yeah.
And one other, in terms of the oysters, I think the people at Bodega have found that the native oyster actually is seriously affected at the settlement
Oh, interesting.
and the juvenile stage
Mhm.
by ocean acidification. I think that's correct.
The Olympia oyster.
Yeah, so they're studying an introduced species, right?
Yes, yes,
So
from Japan.
it could be that that may have some
Yeah.
effect on the results that you got.
Yeah, another thing is the individuals that we use, they came from the molluscan broodstock program, so they've gone through several generations of being selectively bred. And so there are some studies that kind of suggest that this molluscan broodstock program has been inherently selecting these individuals to be more resistant to acidified conditions. So that's something else. I just didn't have time to get into it.
All right, time for one more. If anyone has it. All right. All right.